So, hi everyone, and welcome to the 11th TIA Centre seminar of the academic year. So, for those of you that don't know me, my name's Adam Shepherd, and I'm a postdoc here at TIA Centre. And in these seminars, we try and invite researchers from across the globe to present new and exciting research. So, I'm delighted to introduce Hakim Benkirane today. Hakim is a third-year PhD student working with Central Superlek and Gustave Roussy Institute on applying AI to precision medicine. His field of expertise focuses on conceiving robust and interpretable deep learning-based methods for the integration of multimodal data for precision oncology, with particular emphasis on multi-omics data and histopathology slides. The title of Hakim's talk today is Multimodal CustOmics, a unified and interpretable multitask deep learning framework for multimodal integrative data analysis in oncology. So thank you very much, Hakim, for joining us. We're really excited to hear your talk. And before we get started again, do you mind if so we normally record these talks and put them on YouTube? Is that okay?
Yeah, it's okay. That's fine.
And would you rather we kept the questions to the end, or are you happy for us to ask questions throughout?
Whatever's comfortable with you. I'm okay with both.
Okay, fantastic. Well, if we get any questions as we go along, I'll try and field them. But brilliant.
Of course.
Well, thank you very much, Jen. Feel free to start.
Okay, so thank you for inviting me for this seminar. I will present to you my most recent work about the conception of a unified and interpretable multitask model for multimodal integration for precision oncology. This work is at the crossroad for my work with Central Superlek and Gustave Roussy, and most precisely for the Center of Precision Oncology that's called PRISM. So here I will first start with a brief introduction of the context of the study. Then I will go through different challenges faced by multi-omics integration and multimodal integration that led to the conception of this particular tool. So first, a little bit about the context. It's about using multiple data sources to do precision medicine, precision oncology, so for treatment recommendation, risk evaluation, or for example, patient stratification. So here we have different types of data, going from genomics to histopathology or clinical data. All those different types of data target different levels of the biological system, and there's a huge benefit of being able to integrate all those data because it gives a more comprehensive view of the biological system. And one of the two main challenges faced for being able to integrate those different types of data are, first, to be able to deal with the high heterogeneity between all those type of data, and also being able to interpret the results, because using the deep learning framework for all this huge amount of information tend to sometimes render interpretability a bit more complex. So that's the problem. That's the two main issues that we will try to delve on in this presentation. So first, I will talk about the challenges of multi-omics integration, because multi-omics integration is actually the integration of multiple types of genomic, transcriptomic, or metabolomic data. We have different types of expression going from the gene level to the transcript to the protein, etc. The integration of multi-omics data has been a huge challenge for a long time, because those type of data are really heterogeneous, and the challenge of being is more about how to properly integrate those type of data to perform different different classification tasks like patient stratification or survival analysis, or even do correlation analysis with the different phenotypes, etc. So here the challenge would be to find a good integration strategy that can optimize the most the interactions between the different types of omics data without suffering the huge curse of dimensionality, because here we are at a very high dimensional space. For example, transcriptomics data can go through 12,000 genes. So yeah, there are three main types of integration that are mostly used in the literature for multi-omics integration. We have the three most known integration strategy: early integration, late integration, and joint integration. The idea of those integration being to create a final representation for multi-omics data. So, for example, early integration is the most simple type of integration, as the integration part is done before even plugging into the model. So we have our modalities, we just aggregate them like with concatenation, etc. We plug them into a model, and we have our last representation, our last output. So, for example, in general, for multi-omics integration, we use a variational autoencoder framework to be able to create a final latent representation of those data. So for early integration, it would just be like concatenating genomic, transcriptomics, and metabolomic data and plug them into a variational autoencoder and try to reconstruct the concatenation. So the advantage of this type of method is that, well, it's simple to implement, it's very easy to use, but the problem is that it creates a huge imbalance between the sources. First, because of dimensionality, because, for example, in general, genomic data have around 3,000 to 10,000 genes, whereas methylation can be done in the CPG island, the CPG island level. So if you're using Illuminate 450K, you have 450K features. So there's a huge imbalance because of that. So in general, methylation tends to overshadow the copy numbers. Again, for example, in the quantity of signal, if there's a source that brings a lot more signal than the other, well, we don't have proper interactions between them. So we may overlook some important signal when we're using early integration. The second type is late integration, where each modality is inputted into its own model, and instead of aggregating the input, we aggregate the output. So in the case of variational autoencoder, it would be, well, each source goes through its own variational autoencoder, creates its own latent representation, and we aggregate the latent representations at the end to give a final latent representation. So the benefits of this method is that every source, that it respects the specificities of each modality. Because each modality is trained alone, we can optimize the network for each one of those modalities, making the unimodal representation for each one of them optimal for the modalities. But the problem, again, with that is that it doesn't take into account the interaction between the sources, because we actually don't learn the representation jointly. So yeah, we may overlook some signals that are purely about the interaction between two different sources. And the last type of integration, and the most used for multi-omics integration is the joint integration, where everything is trained under the same model, but instead of concatenating directly the input or directly the output, we try to build a sub-representation per source inside the same model. And the sub-representation are the one that are plugged into the final model. So here, for example, in our variational, it's an autoencoder. Instead of having a single encoder in the same framework, we would have one encoder per source that converge into a central encoder that gives a final latent representation. So, as you may, as it is intuitive to see that the joint representation can correctly model the interaction between modalities as everything is trained into the same framework. But when we started to do experiments using the joint integration for our studies, we found that there's a bit of a problem when used with those particular types of data, and in general in omics data, because they suffer a lot of those type of heterogeneity. It's the fact that all the modalities are integrated the same way, because everything goes through the same model, everything goes through the same loss. We may have problem of respecting each modality specificity. For example, here we in particular tasks, here you can find that we did some experiments of for pan-cancer classification in TCGA with different combination of modalities for joint integration. And we witnessed that here first, the modality that has the highest signal is the RNA-seq, and the highest performance is actually achieved with RNA-seq plus methylation. And when we add copy number variation, it's only worsened the results. Our theory about that is that here, everything is about convergence speed. RNA-seq can converge much faster than copy number variation. And same thing in an extent with methylation. Copy number variation are a more slow signal to pick up. So what happens is that as RNA-seq converge very fast, it sort of dictates its behavior. So when trained on the same network, when we reach convergence of RNA-seq, we kind of reach convergence of the whole network, which makes those kind of results where when we add copy number variation is like we're just adding noise, and the performance worsen. So we wanted the way. Yeah.
May I ask a quick question? Someone in the chats.
Yeah, of course.
So Mohammed's asked, Darwood's asked, how are you handling copy number variation? Are you binarizing?
Sorry, I didn't quite hear the question.
So Darwood's question is how are you handling copy number variation? Is it through binarizing?
No, we don't binarize the copy number. We use them as they are in TCGA.
Brilliant, thank you.
So for proper integration of those type of omics data, we kind of wanted a strategy that is halfway between the late integration that respects the specificities of each modality and the joint integration that's correctly models the interaction between the sources. So we came up with a first strategy that we published in our paper in 2023, which was the first generation of CustOmics, which was published in PLOS Computational Biology, and we called it mixed integration because it's actually a mix between late and joint integration. And the idea here is to do the integration in two parts, where the first part, we train each modality separately to create its own latent representation. So here we have a first part where we go through a variational autoencoder for each one of those of the sources. This by freezing the central encoder. Here, the central encoder in the first part doesn't train; it's completely freezed, so that the first phase acts as a normalizing phase for the omics data, where every modalities have time to converge, and build a robust representation. While in phase 2, then we defreeze the central encoder and we train everything at once as with joint integration. So here we try to mitigate the benefits and the benefits of each one of the latent joint integration without having to deal with the downsides of them. And we tried that with different tasks. The first one was a cancer type classification task where we took the entirety of the pan-cancer cohort of TCGA with 11,000, more than 11,000 patients, and we compared each type of integration strategy, and we showed that our method actually performed better than even the joint integration for variational autoencoder. For this classification task. So here we try to create the latent representation and plug it into a classifier for classifying the different tumor types and normal sample too. When we go through again the different the results for the different combinations of omics data, here we see that we don't have the same problem as before, where RNA-seq and methylation have the highest performance. We can actually achieve this higher performance with copy number, showing that copy number variation still adds information, which is kind of expectable, but it was nice to see that results aligned with the theory. Okay, and because in reality, we never have 11,000 patients in the cohorts, we try to test this method for different smaller cohorts of TCGA. So here for classification, we use the breast cancer cohort to classify the 4 molecular subtypes, and we showed that even with smaller cohorts, our method shows the same behavior as it did with the pan-cancer cohort, which was quite encouraging to be able to use our method even for our cohorts in Gustave Roussy, for example.
Okay, do you mind if we ask a quick question at this stage?
Yeah, of course.
So Mustafa in our group's got a question.
Hakim, can you go back to the last slide, please? Yes. So
this one?
Yes. So you mentioned normal samples here. I might be very naive here. I didn't know there are normal samples in TCGA. If there are, did you consider them as a separate class? Or
yeah.
How did it work?
Yeah, I considered them as a separate class, not.
So separate class per cancer or separate class for all cancers? So like, for example, the site that tissue was taken from, if it was normal for breast and colon, you would consider both of them as normal in a normal group? Or
yeah, I considered both of them as normal. It's normal for all the cancer types, because if I didn't do that, I would have a huge imbalance between the normal samples, because there aren't actually a lot.
Yeah, I was thinking that. Do you have a number in mind? How many total normal samples you had in your experiment?
I don't remember well, but it's it could be counted by I would say 20 or 30, something like that, but
Okay.
really not a lot.
I see. Okay. All right. Just another question. Can you?
Yeah. Sorry, I wanted to ask the arrows in this picture. What do they mean? Or the figure on the right?
The error here, for example.
Yeah. Well, in every square, there's an arrow in every.
Okay, thank you for this question, because I forgot to mention it, because those results have been obtained with the 5-fold cross-validation. So the error is actually the standard deviations across all folds.
Thank you.
And Mustafa, I think you had a follow-up question as well.
Yes. I actually was about to ask quite a similar question. So, how did you average the measures like F1 score or accuracy? Is it like macro averaging or micro averaging?
Actually, what I did is that first the different folds have been done by sampling over the same distribution of tumor types across folds, so that we don't have a huge disparity between the folds. And then I use the macro averaging for F1 score and etc.
It makes sense. Thank you.
Brilliant. Thank you very much. I don't think there's any more questions in the room for now, is there?
Okay, perfect. So now that we went through a little bit of the challenge of multi-omics integration, now we will try to add a bit of difficulty with histopathology data. And I think you're more familiar with the histopathology data, so I won't go through all the details of this modality. But the actual problem is that we have now a high dimensionality for omics data. We have a gigapixel image where we don't know where the signal comes from. And we want to be able to integrate both into the same framework while still keeping the interpretability. And this seemed like some sort of complicated issue, because we have, it's like searching for a needle in a haystack problem, which, so to be able to keep the interpretability aspects of our framework, we envisioned semantic separation, a thematic separation for each modality. So for the histopathology part, when trying to build the histopathology embeddings, we try to do it for each type of region in the histopathology. The region were actually obtained with some sort of soft clustering, then validation of the soft clusters afterwards. So we have one embedding for each type of regions. Then the same thing is done for the omics data. We take different gene sets like pathways, and we divide our input data into different pathways to be able to generate an embedding for each type of pathway. Those embedding are actually performed with the first generation CustOmics to build a robust multi-omics representation for each type of pathway. Then we try to create, as a latent representation, a tensor for which each block represents actually the interaction between one region and one pathway, so that we still conserve this idea of what signal comes from what interaction. So that, for example, if we go through all the representation in this line, it represents the information of one pathway. In this column, for one region. And for each block, we would have a combination of region pathway.
Okay, may I ask a quick question here? Sorry. So how did you go about getting the region level in representation?
The representation is actually, I didn't go through this detail, but it went through a model that I actually conceived in another paper that used a hypergraph representation for the whole slide image, so that we can delimitate the different clusters, and then as everything is trained under the same model for specific tasks, it tries to do the convolution for each one of the hyper edges and have a representation for each region.
Sure. Thank you. And I think we've also got another question in the chat. So Sarah's asked, how did you divide the P pathways? And the second question was just what I've just asked. So
okay. So for the pathways, we can divide them in different methods depending on what biological signal you want. For our experiment, we used the cancer hallmarks that we generally use for gene set enrichment analysis, but you can try the model is actually can take different types of gene sets, so you're not restricted to hallmarks. But for the rest of the experiments, we used cancer hallmarks for the analysis.
Thank you very much, Hakim.
Okay, so then this latent representation would then be used for different prediction, like classification tasks, survival, or clustering. And we would still have like multiple levels of interpretations, going from sort of multimodal pathway enrichment analysis, where we would have a pathway enrichment a gene set enrichment analysis, but with adding the signal of the histopathology, which can quite change the tendency of this gene set enrichment analysis. We would have a gene level importance, signifying what are the actual genes that contributes the most in a specific pathway or in general. And we can have an extension of the pathway enrichment analysis that takes into account also the spatial aspect by adding the information brought by the different regions of the histopathology. So we would have all those kind of interpretations into the same framework. So for the multimodal integration part, the most crucial thing is actually how to compute this block that gives the interaction of the interaction between a pathway and the region. So here we took inspiration from an old paper that did the same thing for visual question and answer with a quite light model. We wanted to keep the model light for better applicability. So the idea was just to use the pooling module introduced in the multimodal factorized bilinear pooling with the co-attention learning for visual question and answering. So it goes through each modality goes through a fully connected layer of their own. Then there's an element-wise multiplication followed by dropout. This is the expand stage of the pooling module. Then for the squeeze stage, we would have just the sum pooling, then two normalization networks. So here we tested for this study. We tested a lot of different types of pooling, and we found that this was the best compromise between between like number of parameters and efficiency. We first started by using like a variational autoencoder for to create this representation, but it was quite overkill for for this application. This simple pooling network actually does the trick. And we don't have to use the co-attention, because here the block is actually self-explaining. Then once we have our different block, our tensor representation, we want to be able to implement interpretability inside the classifier. So we try to use a mixture of expert approach, but to make it more robust, we made it a little bit hierarchical, so that in the first phase of the mixture of expert, we use each line of the tensor to try to optimize the prediction for each pathway independently, so that then we learn the weights of the regions inside the pathway independently, and so that the weights are still robust inside the pathway. Then we would have a representation, like a sub-representation per pathway that will go through the second phase, and then we'll learn each weights for each pathway, so that here we have the weight for the pathways and the weight for the regions, the corresponding regions. And those weights will then be used for the interpretability parts. Okay, so now I will just present briefly the results, how it performs against the state of the art, and do like a small demo of what types of interpretability results it can output. So first, about the classification performance, we tried to keep the same idea as we did in the first generation of CustOmics by using the pan-cancer cohorts for large number of samples. Then by doing the same classification of molecular subtypes for three different TCGA cohort this time: breast cancer, colon, and stomach cancer. And we see here again that our CustOmics framework is able to outperform the state of the art, and this is mainly due to the fact that the multi-omics integration is very performant compared to different multi-omics approach that are used in the literature for multimodal integration, mainly the SNN networks. So for the interpretability part, I will show you the different levels of interpretability that the model can output. So we can start with the pathway level interpretability, where we perform some sort of gene set variation analysis that gives us the importance of each pathway inside for the model. And not only does it give us a pathway importance score, it can also output like a pathway as like a p-value for evaluating the significance of the score, because sometimes you can have like one single gene that goes that has an importance score very high, and the rest of the pathway is at 0, and this gene is actually overlapping with another pathway, so we don't actually output the right importance pathway. But in our setting, we can use as the same vein as in the gene set variation analysis, a differential study that outputs the significance of each pathway enrichment score. So here, for example, for the discrimination between the HR2 versus the rest of the molecular subtypes, we indeed see that this Trojan response is at the top of the pathway importance, which was predictable for breast cancer. So one thing we can do is go a little deeper in the estrogen response and see the gene level interpretability. So here we can try to analyze what actually happens inside the estrogen response pathway, and we can output some gene level importance score. So here, when it goes to the right it means that the importance is tending for the HR2. So here, meaning that TFF a high expression of TFF1 is actually predictive for HR2 and not for the other. And opposite for the blue in the opposite direction. So this is just a way to show how CustOmics can be able to output a gene level interpretability score. And we saw also that those gene level kind of, especially with TFF1, align with the literature for HR2 versus the other the discrimination of HR2 in breast cancer. Sorry. Now about the spatial level. So what, as I said before, we can try to output like a score for the importance of a specific interaction between a pathway and the spatial region for the classification of HR2. So here you can see a quick heat map showing like that the tumoral region is actually highlighted, but we can't say much from this image, as heat maps are sort of rough to interpret. So we did a little analysis of what actually happens inside the high attention regions for all the different patients, and we see that what actually happens is that there's a huge proportion of lymphocytes and tumor infiltrating lymphocytes inside for high attention regions for discriminating the HR2 versus the rest. And the presence of TILs is actually also prognostic for HR2. There are a lot of literature that I specified in my preprint that shows that TILs are actually very correlated with the presence of HR2 expression.
Sorry, Hakim, just a quick question on that. I'm not sure if I missed
of course.
this slide. So the attention score that you get, is this per patch or is it per region for the spatial? Well, when you look at the actual histology images,
it's actually per region.
It's per region.
So it's the score that the mixture of experts gets from here, from this phase. Right. It's the weight for predicting the right output using only the pathways.
Okay, sure, thank you. And I think there's also just a couple of other questions on the chat as well. So Sarah's asked,
of course,
in the methodology, you sort of use the all regions times all pathways and then aggregate them for the final classification. Did you consider attention weighting the fused embeddings?
Yeah, actually, the first idea we had was to use attention weighting for this type of problem, because it's actually what's done in the literature. But as we went through the experiments, we couldn't seem to find like a robust attention weighting. It was always giving us some mixed results, and we weren't really confident with the result that the attention weighting was giving us. Thus, we wanted to have a more robust in framework method for interpretability.
Brilliant, thank you. And we've also got a question from Kava online as well, so asking, considering the different nature of different modalities, do you consider the same embedding dimension for all modalities before integration? And how did you train each embedding?
When you say how did I train each embedding, it's what do you mean?
I'm not sure, Kava, if you're online, whether you can add some more context to your question.
Can you hear me?
Yeah, we can hear you.
Yeah,
okay.
I can hear you fine.
Yeah, thank you. Actually, I mean that when you want to train your model in order to learn the embedding weights, how did you try, for example, for a natural language processing model? I know that for inferring the embeddings, we can use some sort of methods like the Glove-to-Vec and etc. And in order to extract the different vectors for the different words. And in this case, you're using the different, for example, regions for the image and the different pathways for the other modalities. And I wanted to know that if you want to represent a pathway or a tumor region with an embedding, how your model will learn about the different features that you're going to be extracted in that embedding.
Okay, you mean in this part, in the first part, in the modalities
yes,
encoding?
Yeah.
Yeah. So in the modality encoding for our experiment is it's done in multiple phases. So here in the first part, along with the for histopathology, along with the multi-omics data, everything is trained under the same laws. So we train the embedding to be able to classify well the HR2 HR2 against the rest, for example, alone, so that we have specific embedding that can, in a unimodal case, be able to predict our output, like we did in the first generation CustOmics, where we had the first phase that creates a robust embedding for each modality, and we do the same for multi-omics. Then we unfreeze this multimodal fusion model that brings together the two modalities and creates the tensor-like representation. Does it answer your question, or do you have something?
Yeah, thank you very much. Great.
Thanks, Hakim.
Okay, so now we'll just go through a small discussions about the strength of the model and some of its limitations and possible future perspectives. So the main advantage of CustOmics is that it's a light and performance deep learning model that actually competes with the state of the art. It doesn't need a huge visual transformer or a huge LLM to be able to work. It just needs it just needs the graph convolutions and variational autoencoder, which has quite light model in comparison with what the state of the art is actually doing. It is also able to be to perform efficient multi-omics integration through the mixed integration strategy that we came with in our previous paper. It also provides multiple interpretability interpretation tool along the same framework. So for clinical integration, it's very useful to be able to delve through different levels of the biological system, to be able to understand the different interactions between the spatial data and the omics data. If we don't have, for example, technologies for spatial transcriptomics or something of the sort. And another key point is that CustOmics is actually robust to missing data, thanks to to a trick we added from another paper, which is called multimodal dropout, which consists of well, as its name suggests, using dropout for modality to be able to mitigate the effect of missing modalities. So here, for CustOmics framework, we don't need to have all the modalities aligned for each patient. We can have, for example, missing histopathology slides. We can have missing methylations, etc. Some future perspective of CustOmics would be first to add some new interpretability levels. For example, the omics contributions that is not considered in this study. For example, we can see in the future version of CustOmics that can tell us, okay, here for this prediction, the CNV are actually more contributing to the methylation, etc. Here, the histopathology slide is the one bringing the highest contribution, etc. The second point would be to be able to add a self-supervised clustering method to be able to do robust clustering using only the learned representation of CustOmics, the tensor-wise representation, because now if we want to do clustering, we're kind of obliged to see each sub-view of our tensor to see, okay, here we have this pathway that clusters into those type of clusters. Here for the region, we have this, but we don't have a clustering that does the whole latent translation in one go. And we think that with a self-supervised clustering method like deep cluster, if we just add the loss into our multitask framework, we can be able to have robust clustering. And in the end, we would also like to do experiments not only with cancer hallmarks, but with different types of test it with different other gene sets, for example, immunitarial gene set, cancer signatures, etc., to try to extract different biological signals from different gene sets and how they each interact with the histopathology. And we expect that there will be some interesting insights when considering those different types of interactions. So thank you for your attention. If you have any question, feel free to ask.
Brilliant. Well, thank you very much, Hakim, for a really great talk. It's been really interesting. So yeah, we'll open the floor to questions then. Does anyone in the room have any questions they'd like to start with, or anyone online? I know there's a couple of questions online already, so we'll start with those then. So Sarah's asked, did you observe your methods not outperforming for a specific cancer type? Sarah,
indeed.
Sorry.
Indeed, we had a single problem with some survival analysis tasks, especially when not having a lot of multi-omics data. So here, if we when we compared with the state of the art, especially MCATs for whole slide image only, for some survival tasks, we found that MCAT was actually outperforming our model for whole slide image only, and we saw that in multiple cohorts where multi-omics data were far less present than histopathology for specific cases. But in general, it's outperformed the state of the art.
Yeah. So a question for me is really, it's so it seems to me that one of the main advantages of incorporating histology data with the multi-omics is the sort of the high spatial resolution you can get from the histology data.
Yes.
So currently, it's obviously you get like a region level embedding. So how do you envisage improving multi-omics, this model in the future to try and get a more granular attention scores, let's say?
Yeah, this is a very tricky question, because the thing about doing a granular representation is that we have so much batches that we just kind of overfit our attentions to specific cohorts. So here, the first thing we will try to do is try to augment the number of regions considered. So we just augment the number of clusters. For now, the maximum we went through is 10, but we'll try to go deeper and deeper in the number of clusters to see at which point we start having an overfitting on the spatial regions, and at which point it doesn't make any sense anymore. But yeah, this is a nice perspective that we would like to continue to, and which is very hard to envision because of this high resolution of the images.
Yeah, thank you. And do you have any more questions from people in person or online? So Karen online has said, will the PowerPoint be shared? So we will be putting this video on YouTube afterwards, so you'll be able to see the slides there, but I don't know about the individual slides.
Yeah, I can share it with you if you want.
Brilliant, thank you. Fab. And I suppose a final question from me then.
Yeah, of course.
So obviously a lot of the multi-omics that you've been using here is well, I suppose perhaps at the slide level. So how would you envisage using this sort of approach with spatial transcriptomics?
This kind of approach for spatial transcriptomics?
Yeah.
I don't know if it would be really relevant to use them for spatial transcriptomics, as we already have this information. I think we can still try to implement the same multi-level interpretation scores that we implemented in CustOmics for spatial transcriptomics data. But maybe one of the benefits of this kind of method would be to try to integrate spatial transcriptomics with other types of spatial data, like, for example, multiplexed immunofluorescence or spatial proteomics.
Sure.
And try to have like a nice representation for spatial multi-omics data.
Yeah, definitely. That makes perfect sense.
Yeah, but this is a lot more complicated because we're at the cell level and not just the batch level. So yeah, the amount of information is very huge.
Fantastic. Colin's also asked, after asking about the PowerPoint they shared, are you willing to find potential collaborators? So
yeah.
Sorry, sir. I just want to ask, are you willing to find some co-author? Yeah.
Sorry, I didn't quite hear you.
I just want to ask, are you willing to find a co-author, in your research area?
Some cohorts, yeah, we're actually working on some specific cohorts in our hospital. We're trying actually to apply this method on specific internal cohort, but yeah, just hit me up and we can discuss about it afterwards.
Okay, so could you just have your email in the box so that I can send the email to you? Yeah.
Yeah, of course. I will put my email in the chat so that everyone have it.
Yeah. Sorry. I want to make a self-introduction for myself. So I'm first year PhD student from the University of Warwick, and my major is computer sciences. And my research area is the large language model and the graph neural network. So my first publication application is about medical consulting. So the contribution for the first work is use the keyboard retrieval and the emotion understanding to make the consulting generation.
That sounds interesting.
Yeah. I can send you my paper. If you are interested in my first work, and my second work is focused on like the scientific figure generation. So that means I use the LLM to understand a long text and extract the keyword, and then I use the LLM to generate the flowchart like like some very professional flowchart. So then can help our researcher can save many time, and you don't need to spend many time to join the figure again.
Okay, I see. Yeah, I would be glad to discuss with you about that. Send me your paper. Send me your paper by email. I'm interested in reading it.
Yeah. Sure. Yeah. Should I open my video? Oh, sorry, I'm in my home. Yeah.
So, really,
by the way, I didn't send your email address.
Sorry, my chat crashed.
Yeah, I didn't send your email address. Anyway.
Brilliant. Well, if there's no more questions online, then I think we'll draw the seminar to a close. But thank you very much, Hakim. It's been a really interesting talk. I've certainly learned a lot. And just a bit of a reminder to everyone else, we've actually got our next TIA Centre seminar tomorrow at 2:30. So we've got a bit of a bumper week of seminars this week. But we're going to be joined in person by Professor Sabine Taper from University Leuven in Belgium. So hopefully see you all there. But again, thank you very much, Hakim. It's been a really, really interesting talk. And
yeah, thank you.
Yeah, thank you for your today. The seminar is really helpful and really impactful.
Thanks, everyone. Bye, Hakim.
Thanks. Bye.